So in this video, we're going to be talking about fatty acid catabolism in general, lipid metabolism. So this is video 1. So this is lipid metabolism 1. So fatty acid catabolism so to catabolize means to break down. Okay, let's start by defining the word catabolism. So it simply means the breakdown of triglycerol. Okay, so breakdown of triacylglycerides. Okay, now they're broken down to carbon dioxide as well as water in order to produce energy, which is ATP. And the overall process of this is what we call beta-oxidation. Okay, so this is what simply talking about how fats are broken down in order to give the body energy. Having said that, fatty acid oxidation can provide approximately about 80% of the energy that's needed for certain tissues like the heart, the liver, as well as resting muscle tissues. Okay, so these are the organs that are supplied by energy. Okay, so fatty acid chains, when they get oxidized to water and carbon dioxide, the electrons have to pass through the oxidation reduction chain, which is the electron transport chain. Okay, the ETC. Alright, so triglycerides actually produce more energy because they are quite highly reduced. Okay, so the more highly reduced the compound is, the more it's capable of producing more energy as compared to glucose. So these triglycerides, they produce about 2 times amount of ATP as compared to glucose. Alright, this is because they are highly reduced. That's the reason. So why exactly does the body store fats in the first place? So we're going to learn that there is no limit. So there are 3 points. Point 1 is there is no limit to storage of fats. Alright. Second point is that because fats are hydrophobic, they do not affect the osmolarity. They don't affect the osmolarity of the cell. Which is quite very important because they will not affect the concentration gradient within the cell. And the third point is that fats are innate, therefore they are not likely to react or rather harm the organism because they are innate. Alright, next thing that we're going to learn is storage of glycogen. So glycogen is stored in small amounts, for instance the liver as well as the muscles. However, glycogen provides energy for up to about a day or so, which is 24 hours. And glycogen is also polar. Alright? Glycogen is polar. This is to tell you that it does affect the osmolarity of the cell. Okay? So, fatty substances are 1, hydrophobic. They do not interact with water, or rather they are water hating. 2, the metabolic enzymes are water soluble. So the enzymes involved are water soluble. Okay, hence they can dissolve in water. Then the digestion of fats requires certain features that help these fats to be more susceptible to these enzymes. Okay, they have to be broken down before they can actually be worked on by those enzymes. Okay, so this is to say, next up we are going to look at the digestion of fats. How are fats digested? Okay, so we have what we call bile salts in the body, right? These bile salts are amphipathic. Alright, so they interact with insoluble fat granules. These are the fat granules. They convert them into dispersed micelles. Okay, you know that a fat is found in a micelle like this, right? So what this bile salts does is that it cleaves those ends and enables them to be separate, so they'll be dispersed micelles, and by so doing it increases the contact between these fatty droplets and the enzymes that work on the fats. Alright, so the liver makes up bile from cholesterol. So bile is made from an element called cholesterol, and this bile is formed in the gallbladder. So let's take, for instance, is our liver. There is what we call the gallbladder, which is there. It has certain other features. So it goes to the pancreas. There's a tube that goes to the pancreas as it is going to the duodenum, and so on and so forth. Alright, so what is happening is that the liver stores bile in what we call the gallbladder. Okay, the gallbladder is responsible for storage of this bile, and it only releases it into the small intestine after someone has taken a fatty meal. Okay, or rather a meal that's full of fats. So bile acids so what the bile does is that it acts like a detergent, like a soap. So what it does is that it breaks these fats into smaller pieces, or rather smaller chunks. So if the fat was this big, it will break it down into smaller pieces so that those enzymes are able to work on the fats. So like we said, the enzymes are water soluble, and one of the enzymes that we're going to talk about is lipase. So lipase is now able to interact with those fats that underwent, sorry, didn't mention this, but they undergo emulsification, which is the breaking down of fats into smaller chunks. Okay, and what does that is bile. So the water soluble lipase is now able to interact with the lipid or rather the micelles, and this is what converts the lipid into triacylglycerides. Okay, so it converts these triacylglycerides into monoglycerides. Okay, diglycerides, meaning two. So in short, this is diglycerol, sorry, as well as fatty chains. This is just to say that the lipase is just converting these triglycerols into simpler molecules which the body is able to absorb into the blood. So these products then diffuse into the intestine. Okay, so this is our intestine, right? Those products are now able to diffuse within the intestine, and remember that this lipase is actually produced by the pancreas. So all this breaking down is happening in the pancreas, and then after it goes in the intestines right next. Okay, so as they are released in the intestinal epithelial cells, they are reformed. Okay, so when they are released here, they reform back to triacylglycerol. They form back into that form through which they were broken down. Okay, so they form down, they are reformed, creating the triacylglycerol, and then they're further packaged with cholesterol. So this is packaged with cholesterol and specific proteins plus these proteins, and when this happens they form what we call chylomicrons. Okay, they form what we call the chylomicrons. So these specific proteins that we were talking about, we call them the apolipoprotein. Apolipoproteins. Okay, those are the special proteins. So those apolipoproteins bind to the triglyceride, the cholesterol complex, and these are what help to transport the lipids into various organs. Okay, so this is what's going to happen. I'll draw just a quick diagram so that you're able to understand what I'm talking about. So the protein component of the lipoprotein, right, in the chylomicrons, it serves as a ligand. Okay, and we know that the ligand enables it's like a receptor. It enables the picking up of signals when there is a hormone that's available. So these ligands the ligands serve as the chylomicrons serve as ligands, which the cell receptors are able to recognize. Okay, so there's recognition due to these ligands, and they aid the movement of the chylomicrons within the intestinal wall. Okay, they enable these chylomicrons to move. All right, so let's just go straight into looking at the structure of a chylomicron. Okay, so this is just a brief structure of the chylomicron. It has phospholipids, then these three are the apolipoproteins. All right, then these are the phospholipids, and then this is the cholesterol that's embedded. So that's the cholesterol. Okay, so the surface of this chylomicron, this whole thing is the chylomicron. The surface of the chylomicron is made up of the phosphodiester head groups of the triglycerides, these that were able to see. So the fatty acid chains face inwards, which are these face inwards, whereas the aqueous part of the of this phospholipid. Okay, so the phospholipid has two parts. There's one that's water loving and there's one that's water hating. That's what determines where these actually face. Alright, so the next thing that we're going to talk about is digestion of fats. Okay, so in the digestion, what you learn is that apolipoprotein C. Okay, so let me just denote it as APC2. So this is apolipoprotein C2. Okay, what it does is that it activates the lipase. This one will activate the enzyme, the lipoprotein lipase, which will then break down the triglycerol to fatty acids and glycerol. This is your fatty acid. So this lipase will now break down the triglycerol to fatty acid and glycerol. Okay, so the first thing we talked about is the apolipoprotein C2 will activate the lipoprotein lipase. This is the enzyme. The enzyme will then break the triacylglycerol, okay, into the fatty acid, okay, and the glycerol. After these two are after these two are formed, the fatty acid and the glycerol, they are taken to their target tissues depending on the energy need that that tissue has. If it's the let's take for instance, if it's the muscles, they need energy, then that's where the fats are going to be channeled. All right. So in muscles, the triglycerides are oxidized to release energy when the person is starving or doing a lot of exercises that demand lots of energy. Fats are usually used as an alternative. Okay. Then we also talk about the adipose tissue. Okay, or rather a fat tissue. So we have the adipose tissue. In the adipose tissue, it's stored as fat reserves. All right. So these store fats. All right. So in the fully fed state, the body is going to take up those fats and store them in the adipose tissue. All right. However, fats are also stored in cells that synthesize steroid hormones. Okay, cells that synthesize steroid hormones, like the ovaries, the testes, as well as the adrenal cortex. So you also find fats in these fatty acids and glycerol in these organs, because this is where there is steroid hormone synthesis. Okay, so next up we are going to talk about mobilization of fatty acids. Okay, mobilization of fatty acids. So under this, let me just draw something real quick. So firstly, there's an issue of hormones. Of course, hormones always work with the receptor. Then there are what we call G5 coupled proteins, and these form what we call cyclic AMP with the use of ATP. Okay, so we have cyclic AMP, and of course this is the enzyme adenyl cyclase. Okay, so what is happening is that lipid droplets are usually coated with what we call perilipins. So these are the perilipins. Perilipins, they prevent fats from being digested before the actual time, or rather they prevent fats from being used at just any time. Okay, so they help to store those lipid droplets. So hormones are the ones that signal the release of fatty acids by use of metabolic active tissues like the skeletal muscles, the heart, and the kidney. So to be specific, the hormones that are involved are epinephrine and glucagon. Okay, so not only does glucagon enable the release of glycogen to give us glucose, but it also enables these fatty acids to be broken down in order to release energy. So what these hormones do, like I had drawn here, let me use a different color. So what the hormones do is that they trigger the formation of cyclic AMP. Okay, this is ATP. ATP is used to make cyclic AMP by the use of adenyl cyclase. So in response to the cyclic AMP, these perilipins that are drawn here, these are the perilipins. The perilipins are the protein. These are the proteins that are stored on the fatty acid droplets. Okay, so what happens is that they get phosphorylated by a cyclic AMP dependent kinase, which is pKa. So pKa goes to phosphorylate. Okay, let's just erase some things. So so we have our pKa right here, right? This is pKa that's dependent on camp. So pKa phosphorylates the perilipins, and when the perilipins get phosphorylated, they can now interact with the hormone sensitive lipase. Okay, when they interact with this lipase within the site or so, this is what causes the hydrolysis of these fats. Okay, it means they are able to now give way for the lipase to just break down those fats into fatty acids and glycerols. Okay, so next up is we are still talking about mobilization of fatty acids. So the action of these fatty acid sorry, just a minute. So the action of these fatty acid of this lipase releases the free fatty acids. Okay, so there's a release of free fatty acids from the adipocyte into the blood, and as many as 10 free fatty acids can bind to serum albumin in the blood, and they get transported to the heart. Okay, so let's go back to the diagram so that you can see. So for instance, this is our blood, right? There is a serum albumin that we have. So what happens that when these fatty acids are released, they bind to albumin, and that's how they are transported in the blood. Alright, so they are transported in the blood, and they are also transported to other parts like the heart, the skeletal muscle, as well as the renal cortex. Alright, so this is the way of transporting an insoluble molecule within an aqueous medium when it binds to this serum albumin. Okay, it binds to the serum albumin, and then it's transported to organs that are in need of that fat. Alright, next up we are going to talk about the transportation of fatty acids into the mitochondria. Okay, so let's write it down. So this is transportation of fatty acids into mitochondria. Okay, so enzymes that are involved in the beta oxidation reduction, they are found in what we call the mitochondrial matrix. That's where the enzymes are found. So the free fatty acids that have less than 14 carbons, these are the ones that enter the mitochondria through what we call protein pouring channels. Alright, so as they enter, so this is the medium they use. However, the free fatty acids that have like greater than 14 carbon atoms, or a majority of those fatty acids, these ones require a special transporter. Okay, in order for them to enter the mitochondrial matrix. Alright, so this occurs in 3 enzymatic steps of the carnitine shuttle. Alright, so reaction 1 is that the fatty acid, so we're going to understand this reaction by writing a chemical equation. So the fatty acid is added onto a CoA group, an CoA group, and this is catalyzed by acyl synthesis, acyl CoA synthesis. So ATP is released. So this is going to give us fatty acyl CoA plus AMP plus pyrophosphate. Okay, so the first thing is that the fatty acid is added to CoA, and the enzyme that aids this is acyl-CoA synthetase. Okay, that's the enzyme. So this is reaction 1. Then reaction 2, the fatty acyl carnitine passes through the intermembrane space. So this is our fatty acyl component. It passes through the intermembrane space and into the mitochondria complex matrix. So this is our mitochondrial matrix, right? So this passes through and goes to the mitochondrial complex, and it doesn't just go through just like that. It uses an acyl carnitine transporter, all right, in the membrane. Then stage 3, or rather reaction 3, is that while in the mitochondrial matrix, all right, we have our mitochondrial matrix. What happens is that carnitine acyl transfer 2 is the one that transfers the fatty acyl group back to coenzyme A. Thereby it recreates the fatty acyl CoA molecule. This is what's happening. Okay, maybe let me write it down. We have what we call carnitine acyl transferase 2. So this is one that transfers the fatty acyl group, okay, back to coenzyme A, and this is what to recreate the fatty acyl CoA molecule. All right. So there are certain disorders that are related to the carnitine shuttle. Okay. Firstly, the first disorder is related to carnitine palmitoyl transferase deficiency. So let's just label it as deficiency. One is hypoketonic hypoglycemia. Okay, we also have liver dysfunction, or which this one results in liver damage, or rather liver failure. Okay, then the other type of deficiency is the carnitine acyl translocase deficiency. Okay, this is the other type of deficiency. So this name is deficiency 2. This one brings about cardiomyopathy. The second one brings about hypoglycemia. The third one, or the third disorder, sorry, is sudden death. The fourth one is hyperammonemia. Sorry, don't mind the handwriting. So these are the disorders that are involved. Okay. Next up, we are going to talk about the 3 steps of beta oxidation. Okay. So in step 1, so this is beta oxidation. Step 1, 2 carbons are successfully removed to form acetyl-CoA. Okay, you have your carbon chain. All right, this is your carbon chain that's highly reduced, and so on and so forth. So at the end, you know that there is a carboxyl group, right? So what happens is that 2 carbons are removed. 2 carbons are going to be removed in order to form the acetyl-CoA, and these are removed starting from the COO end. That's where the two carbons are first removed. So the first enzyme has three isoenzymes. The first enzyme that's used to remove, it has what we call three isozymes that act on the 4 to 8 carbons. So you know that this is a long chain of carbon. The first three enzymes will act from carbon number, from what can I say, from about 4 to 8 carbons, then they act on 4 to 14 carbons, then 12 to 18 carbons. So this is to tell you that if you have a 16 carbon fatty acid, right? This fatty acid is able to yield about 8 acetyl-CoA molecules, because remember it's removing carbon atoms 2 by 2. So when you divide 16 carbons divided by 2, it's going to give us 8 acetyl-CoA molecules. Okay, hence therefore it passes through this process about 7 times, because remember the fatty acid is very long. It just doesn't have about 16. There could be more. Alright, so in these processes the NADH and FADH2 are produced in every cycle. These are the products of beta oxidation. Alright, so in beta oxidation there are two types of enzymes that catalyze the last three reactions. So the exact enzyme depends on the type of fatty acid chain that's being worked on. Okay, these are the two types of enzymes that are worked on, that are involved in beta oxidation. So if the carbon atom has 12 plus number of carbons, okay, it's worked on by a multi-enzyme that we call trifunctional protein. So this is the trifunctional protein. Okay, this trifunctional protein is made up of 8 chains. 8 chains which include 4 alpha as well as 4 beta chains. So this trifunctional protein is the one that's involved in the beta oxidation reduction of a fatty acid that contains about 12 plus 12 or more carbon, 12 or more fatty acids that have, 12 carbon fatty acid. Yes, that's rightly said. So the alpha chains have what we call the beta CoA hydratase. Okay, and the beta hydroxy acyl CoA dehydrogenase active sites. So if these are active sites, it means these are specific sites through which these enzymes that have been mentioned, that's why they bind. Alright, however, this is for the alpha, but when you come to the beta chains, they have a thiolase active sites. Okay, so the alpha have beta-NO-CoA hydratase and beta hydroacyl CoA dehydrogenase active sites, whereas the beta chains have thiolase active sites. Alright, so the fatty acid chain that has 12 carbons or shorter, This one is worked on by single enzymes, unlike that one that's worked on by trifunctional enzymes. So this one is worked on by single enzymes that are dissolved within the matrix. Alright, the next step is beta oxidation stage 2. So in stage 2, remember in stage 1 we had to produce acetyl CoA. Now in stage 2, the acetyl CoA units that are produced, they go in the TCA cycle or the tricarboxylic cycle or the citric acid cycle, and when they go in this cycle produces NADH as well as FADH. Alright, that's what happens in step 2. However, stage 3, the FADH2 and NADH, these are channeled. They are transported into the electron transport chain in order to produce ATP. Alright, so it's a series of steps. They pass through the electron transport chain in the mitochondrial matrix, and this is what produces ATP. So this is the flow of steps that goes on. So how does what are the results of the beta oxidation, and what's the energy yield? So let's just label this as energy yield. So under energy yield, each FADH donates 1 electron. This one is able to donate 1 electron to the electron transport chain. Okay, now when this happens, 1.5 ATP molecules are formed. So for every 1 electron that's donated by FADH in the electron transport chain, 1.5 ATP is produced. However, 2.5 ATP molecules are yielded when NADH is the one that is donating the electrons. So there is a total of 4 ATP molecules per cycle. So per cycle there is a total of 4 ATP. For every cycle of the fatty acid oxidation, we have 4 ATP molecules that are produced. So even from this you can tell that there's a difference. That's why we said fatty acid oxidation yields about twice the number of ATP molecules that would be yielded when glycerol is broken down, because when you look at glycolysis, we only have a total of 2 ATP molecules that are formed at the end of the day, but for fatty acid or rather beta oxidation, there are 4 ATP molecules that are formed in contrast. Okay, so this is what is happening. So this is the equation, one of the equations that's happening in the oxidation reduction, where NADH is undergoing oxidation. It gives us NAD plus. It has lost an electron, plus water. So now let's consider the energy yield per molecule of acetyl CoA. So we had talked about those FADH and NADH, but now we are talking about acetyl CoA. So this acetyl CoA, when it's produced in the beta oxidation reduction, it enters the citric acid cycle, right? The citric acid cycle. It enters the citric acid cycle, and in this citric acid cycle we have 1 molecule of FADH, but we have 3 molecules of NADH2 that are actually produced, and automatically from this you are able to calculate the number of ATP molecules that are going to be produced after these molecules enter the electron transport chain. Alright, so this is about acetyl-CoA. Now we are going to talk about what we call ketone bodies. Okay, what exactly are ketone bodies? So, ketone bodies are formed in the liver as well as the brain. So the brain can use these ketone bodies as an alternative source whenever we don't have glucose in the body. It can't use fatty acids. Okay, it can only use ketone bodies or glucose. So all the organs except the liver, which lack enzymes to convert them to acetyl-CoA, are able to use this fuel. Alright, so these ketone bodies comprise of three components that we call acetoacetate. So we have acetoacetate, we have acetone, and beta hydroxybutyrate. We have beta hydroxybutyrate. So these are the three components of the ketone bodies. However, when you look at acetone is volatile, meaning it can easily evaporate, and because of that, when ketone bodies are formed, you can detect the acetone in the breath. When someone breathes out, you're able to detect acetone. Alright, sometimes we use this test to diagnose for diabetes. Okay, meaning the body is using up fatty acids instead of glycerol. So this means that this is a problem. Alright, so when a person is starving of carbohydrates, okay, or when the carbohydrates stores are depleted, the body will rely on gluconeogenesis. Okay, to rely on gluconeogenesis as well as the citric acid cycle. Yeah, so those are the ones that are relied on. So the citric acid cycle has intermediates, or rather has those components in it that can be channeled into the production of sugar. However, it's important to remember that the organs such as the brain that we've been talking about, the brain requires glucose as its primary source of energy. Alright, so to free up more of that coenzyme in cases where the body is starved of carbohydrates, the acetyl-CoA has to be converted to what we call the ketone bodies. Okay, so in untreated diabetes mellitus, okay, diabetes mellitus is where the person has low insulin levels. So this one means that glucose entering the body after a meal cannot be properly absorbed and stored as a fuel. Alright, because insulin is the one that's being used to store that, to convert that glucose to glycogen. But if it's low, this means that this person will probably have signs of glucose, maybe in their urine or their sweat. Okay, so once again, the citric acid cycles intermediates, they get channeled into the gluconeogenesis like oxaloacetate, but at the same time, more fatty acids they enter the mitochondria. Okay, of course, through that carnitine shuttle during starvation when there is a low energy state. So beta-oxidation proceeds so as to make acetyl-CoA. Okay, now acetyl-CoA, when it's being produced, has nowhere to go because the citric acid cycle intermediates are going to the gluconeogenesis, so it will not work effectively to give the energy. That's why this acetyl-CoA has to be converted to ketone bodies that the body can actually use to give us energy. or in specific the brain. So these ketone bodies are usually acidic in nature. They are acidic in nature, so the blood pH reduces, and this causes acidosis. All right. So too much of ketone bodies can lead to eventual death or comatosis, or a comatose state where someone goes in a coma because now they don't have enough glucose. So this is it about lipid metabolism. This is lesson 1. Hope you enjoyed the lesson.